So, you're probably wondering, how in the world are we going to identify sharks using just DNA? By using the lab technique, gel electrophoresis. What is gel electrophoresis? Gel electrophoresis is a laboratory method that uses electricity to separate mixtures of DNA, RNA, or protein. In our case, shark DNA. Once we load our DNA samples into the wells of a gel, the gel is placed into the electrophoresis chamber, where an electrical current is run through the gel. Running electricity through the gel causes differently sized DNA fragments to separate. We can then observe how our mystery shark DNA moves in comparison to our two control samples to identify it. Let's take a look. First, we need a gel like the one you loaded. To make this gel, we mix agarose powder with a buffer solution, and then we heat that mixture until all the powder is dissolved. This hot liquid is poured into gel molds that contain plastic cones. Very similar to Jell-O, this liquid solidifies at room temperature. Then the comb is removed and we can see the wells all along the top of the gel. Next, we need our samples. Let's think about DNA. DNA is structured as a double helix, or two paired strands of nucleotide bases. All sharks have some pattern of those bases in common, and specific species of sharks also have their own unique sequences. By using PCR, a sample of each of these DNA sequences has been isolated for this lab. PCR stands for polymerase chain reaction and works by amplifying a specific region of DNA. This causes the DNA to make millions of copies of itself, ending up with a sample of DNA that is large enough for us to be able to use in electrophoresis. So let's get started. First, we'll load our poor beagle DNA into well number 1. We will use the poor beagle control to represent species that are not great white sharks. Then, in well number 2, we will put our second control sample, which contains great white shark DNA. Then, in well number 3, we'll load our mystery sample of shark DNA, which will be identified by comparing the two control samples. On the top of the chamber is a black wire, the universal color for a wire providing a negative charge. And at the bottom of the chamber is a red wire, which represents a positive charge. When we close the lid, the circuit is complete and we can run an electrical current through the chamber. Now remember, DNA has a negative charge because of the phosphate backbone. Now think about what you know about electricity and predict which end of the chamber the negatively charged DNA will be attracted to. The DNA will be attracted to the positive end of the chamber because opposite charges attract. This causes the DNA to migrate through the gel. So, why is that important? How will the shark DNA separate if they're all migrating towards the same end of the gel? The answer to this has to do with the structure of DNA. There are 4 nucleotide bases: adenine, guanine, thymine, and cytosine. Each nucleotide pairs with another to create base pairs and creates the double helix shape of DNA that you are used to seeing. Base pairs, or BP, is the unit to measure the length of DNA fragments. So the more base pairs a fragment has, the longer that fragment is. In this lab, we use PCR to help us amplify 2 specific fragments of DNA from 2 species of sharks. The 2 DNA fragments that we're looking at, remember, the one that is found in all species of sharks and the one that is specific to great whites, have distinct lengths. Longer DNA fragments will move more slowly through the gel because they have a harder time moving through the pores. Shorter DNA fragments will be able to travel through the gel's pores much more easily, and as a result, move faster. So, in well number 1, where we put our poor beagle control, we will see our ribosomal DNA that is present in all sharks. This fragment is 1,340 base pairs long. In well number 2, we will see the great white shark specific fragment, which is 580 base pairs long. But remember, our great white shark is a shark, so it will also contain the ribosomal DNA fragment. Think about what we just discussed. Which sample can we expect to move faster through the gel? The great white specific fragment will move faster through the gel because it's shorter. By comparing our mystery shark sample to our control samples, how can we identify the mystery sample? If 2 lines are present, what would that mean? If no lines are present, what would that mean for our mystery sample? So let's get down to business and send an electric charge through this gel. Once enough time has passed in the electrophoresis chamber, you'll be able to identify your results.